Greetings, everyone. Welcome to the Project AP Research Roundtable. I am Bina Manyar, founder, and it's good to have folks on here. I thought because it's a small group today, this will be shared on our YouTube channel. It would be nice to just, if you don't mind unmuting yourself, to just do a brief intro, your name, your kind of area of interest towards AP. I think that might set a really nice stage for two of our presenters today, as well as a discussion, which we should have time for. Perhaps we can go around. I'll nominate the first person that I see, Dr. Dennis Lal, and then Dennis, after you go, you can just pick somebody else on Hollywood Squares.
Okay, so yeah, hi, I'm Dennis. My interests are genotype-phenotype association, so different types of genetics lead to related or similar disorders, data sharing to online platforms, and genetic counseling and specialty clinics clinically. And Tobias can go ahead next.
All right. Yeah, I'm Tobias. I'm a member of Dennis' team. So that said, my interests are really shared with Dennis. So I'm looking into genotype-phenotype correlations in monogenic disorders, but also across CNVs, which affect multiple genes, and work with online platforms to facilitate the data and make the data accessible to a larger community. Yeah, Kumara, why don't you go ahead?
Hi everyone, I'm Kumara. I'm a postdoc in the ECRA lab at the University of Washington. My work is focused on trying to understand structural variation and how they impact human health and evolution. And I'm particularly focused on using new methods like long-read sequencing and genome assembly to try and study these variations. And I'll let Jen go next.
I am Jennifer Whitmire, an assistant professor at Elizabethtown College, and I'm interested in looking at structural MRI data. So we're looking to, one, be the first to quantify gray matter structure, cortical thickness, gray matter volume in certain brain regions with certain phenotypic information. And we're also working towards creating a kind of like an atlas for individuals with 8p chromosomal disorders. And Nicoletta?
Hi, I'm Nicoletta Moissoy. I'm professor of cellular signaling and pharmacology at the Montfort University in Leicester, UK, and our work on the 8p project has been focused on phenotyping mitochondrial nucleus communication in fibroblasts, basically. Laura?
Hi, I'm Laura Ruth-Bach. I'm the co-president of the Jane Foundation. The family that started the foundation has an interest in AP rearrangements.
Or do you want to nominate somebody?
Sorry. I'll nominate Christy.
Hi, sorry, I had my camera off. I'm Christy Malik. I'm at Children's Colorado, and I am doing like clinical research, heading some clinical research there. So I'm doing some data analysis from the multidisciplinary clinic we have here, along with Dr. Demarest and a bunch of other people. So, and then I will be honest, I don't remember who's still left at this point.
Oh, that's okay. How about Aaliyah?
Hi, I'm Aaliyah. I am recently a graduate from my undergrad in neuroscience. I actually was working in Dr. Whitmire's lab and started working through AP as an internship. But again, kind of in correspondence, we've been really focused on looking at MRI structures and like kind of figuring out how to quantify what we're looking at to be able to really put something together in the future that might be beneficial.
Fred?
Got my camera working just on time. Yeah. Yeah, I'm Frederick. I'm the CSO at Unravel Bioscience. So we work with Bina and the 8P heroes. We did swabbing, produced RNA sequencing data, did the drug predictions, and there's still ongoing work to analyze like on like the foundations and meeting with Lauren to see how we go from those prediction and the various like genotypes and phenotypes. Figure out how we reduce that to a more easier to handle a list of drugs to test in other models. Or I mean, models are tough to do for 8P, but you know they're not undoable. They're doable. Like it's not impossible. That's the idea. So yeah, nice to meet everyone. Yeah, I mean we can name. I don't know. Is Nicoletta? Did she?
She did, yeah. Ethan can go. And also, Fred and his team at Unravel will be presenting next Monday on the actual ways that they looked at the RNA-seq data from our community of patients and controls, as well as the shorter list, still a very long list of repurposing small molecules that are a combination of over-the-counter, et cetera. So that'll be next Monday, which I think will be a really interesting conversation. And I think right now on the call, the only physician on the team is Dr. Malik. So hopefully, Dr. Malik, you and the team can be on that too, because we'll need our doctors who have actually seen and taken clinical care of our AP heroes on that meeting. So Ethan, Katie, or Jacob, maybe the internal team, somebody. Okay.
I'm Ethan. I was serving as Project AP Science Director for several years, and last year transitioned to the Medical and Scientific Advisory Board, and grateful to see all the familiar faces and the new faces.
Jacob.
Everyone. I work at 8P as a data analyst, so I'm in charge of organizing the data, making data dictionaries, making it more legible for data sharing, basically.
Jacob's intimately involved in all of our data, so probably has the precious gold of our community in his hands, and he takes good care of it. Katie, if you can't unmute, it's okay. I can kind of, many of you probably know Katie Severson. She's an 8P caregiver. This is near and dear to her heart, as is mine, and Katie is handling a lot of operations and patient engagement for our natural history study and biorepository. So can't do any of this without Katie. Anuj?
Hey guys, good afternoon. Sorry for running a few minutes late.
Oh, hey. Okay, another physician on the call. Wonderful.
My name's Anuj Jayakar. I'm a child neurologist at Nicholas Children's Hospital in Miami. I've taken care of a couple patients with AP in the past, but have one that I've been talking with Bina about more recently.
Thanks for joining us. Okay, so just to make sure, I believe everyone went that's in here, which is great. So we'll jump right in. Dr. Kumara Masarosa is again, as you mentioned, out of Evan Eichler's lab. We had initially engaged Dr. Eichler's lab in doing some work to really understand the structural variation and the nuances of 8P in and outside the CNV, and had some interesting discovery on that. One of our first funded projects when we had launched the foundation. So Kumar will be presenting and talking about some of his intentions of further looking at the breakpoint characterization in our families, and maybe some of the other work that he's done outside of AP. So without further ado, Kumar.
Okay, thanks, Bina. Let me share my screen. Just 1 second, battling with the multiple screens.
Sure, and Kumar, are you comfortable if like people just chime in and raise their hand if they have questions throughout your presentation, or do you want to wait? Okay, great.
Both, whatever is more is preferred. I'm okay stopping here and talking about it.
Thank you.
Okay. Here we go. Hi everyone. As Bina said, I'm Kumara Mastros. I'm a postdoc. Today I'm going to talk about what's our plan to characterize complex 8P structural variation and try to identify variations that are clinically relevant for the 8P heroes. Our lab has focused for probably in the past 5 years a lot on long-read sequencing, but we did work in the past with short sequencing a lot. And short-read sequencing is great because it's cheap, is very good for large-scale project, point variation, and non-repetitive regions. But it comes to a problem when we have a situation like a duplication. If the reads are too short, they can mismap, and so we are not able to have high specificity. In this case, long read sequencing helps a lot because these reads can traverse multiple complex regions and map unambiguously. The only problem is that they are more expensive, even if in the past 5 years we had a decrease of almost 10x in the cost. But ultimately, we think they're going to be the ultimate test because they are very good at calling any type of variance, really. And long-read sequencing and their data can be used in two ways. The first one is to basically, is shown here, is basically treat them as short reads. So we align them to a reference genome and we look for signature of a structural variation, like in this case, for a deletion and duplication, another complex one. The one that we aim to move to is genome assembly. And it's something that I'm more and more interested with because the idea is to use this data to reconstruct the entire genome of an individual and so have the complete sequence of all the chromosomes and look at pretty much any region of the genome. And the classical situation in a family is that we can then compare the child assembly to the parental sample, and just comparing the sequence, we can identify all sorts of variations. And this is basically would be a high quality assembly comparable to a reference genome even. So I put this example here. This is from Hyatt et al. 2021. The authors of this paper described this case of a child with developmental delays, and they identified this complex variation on chromosome 6. It's basically is a pericentric de novo inversion with a rearrangement of all these fragments over here. And then they found another complex variance that involved an exchange of material between chromosome 7 and chromosome 9. And so they eventually concluded that this one below was the causative variant, but they also report that several other genes were disrupted by this variation on chromosome 6, and some of them were intolerant to loss of function mutation. So this is one of the classical cases where this complex variation could contribute to the phenotype of the child. So the idea is to do a similar work now that all the methods that we have are fully developed. And some of you might know Glenys Logston. Glenys was a postdoc in our lab. She's now at the University of Pennsylvania. And she started this project because she was very interested in chromosome 8, which contains several structures that are interested for the human genome, but particularly the beta defensive locus, which is one of the most complex region of the human genome. And the beta defensive locus is known because carries a lot of repeats that mediate rearrangement, including inversion, duplication, and deletion. And so Glenys collaborated initially with Bina and her family, trying to get high molecular weight DNA that was extracted from blood and cell lines. And she sequenced using PacBio, iFi, ONT, and Illumina, the genome of all Bina's family. And here I'm showing you actually one of the genome. This is from, and you can see how complete the genome is. If you look at the table here, you can consider a human genome as two copies, one from the mother and one from the father of around 3.2 gigabases. So you can see that here we're representing pretty much the entire genome. And you can also see that several chromosomes were complete. This means that we had a unique sequence that went from the telomere to the other telomere. And they are indicated here as an asterisk. You can see how complete these are across these chromosomes. So the first analysis that Glenis did was compare maternal haplotype, maternally inherited haplotype, and paternally inherited haplotype for chromosome 8. And she noticed that the maternal chromosome 8 had an additional sequence compared to this allele. And we really validate this structure that we assemble. One of the classical method is to realign long reads to the assembly and show that we have uniform coverage. When we do not have uniform coverage, that usually indicates a collapse of a sequence and misjoin on any other problem in the genome assembly. But we do a lot of checks to be sure that what we assemble is correct and representative of the genome. The second thing that Glenis did was compare the Carina chromosome 8 to the maternal one. And here we can clearly see what the rearrangement is. It's basically an inverted duplication of this fragment and then a loss of a fragment here at the telomere of chromosome 8. So it's a deletion paired with an inverted duplication. And you can see here that we also have the breakpoints that fall into this gene. It's not really known what this gene do. These are RNA gene, and it's unclear their exact function. But we could find exactly this breakpoint just realigning the long reads to the maternal assembly. And look where the reads break. You can see here that for one of the allele, we have these reads that are breaking, indicating that they map, the rest map somewhere else. And that's how we could identify the breakpoints. Similar on the other side, this is the inversion. But also to be sure, we also did a validation. We asked some of our collaborators in Italy, Mario and Flavia, to run a fish experiment. And you can see here at the top where the probes were designed. And so this way, they were able to confirm both the deletion and the inversion, as well as the breakpoints. Another analysis, these are new data that were generated very recently, is we wondered how this structure compared to the general population. So now we have access to a large number of genome assemblies from the HPRC and HSVC consortia. We are in around 200, 300 genomes from the general population. And another former postdoc of the lab, David Porubsky, did this analysis. He basically looked at the 8p23.1 locus in all these samples. And here I'm representing with this color all the repeats in that region. So sequences of the same color will be highly identical. And we call this segmental duplication block A and segmental duplication block B. Here at the side, you can see how many people carry this haplotype. We have 19 representative haplotypes in total. And you can see that some are much more common than the other. These are number of haplotypes carrying this exact configuration. And also they are colored based on their ethnicity. So you can see that there are even some that were just present in one individual and that are very, very rare. So we looked at how they compare this to the first family that we looked at. And you can see that we separated block A and block B because sometimes the configuration can be a mix of the two. And you can see that proband H1 and mother H1, as well as maternal H2, are very rare compared to the general population. And some of them were only found in the African population, which is known to have more variation. So the last thing that we did on this part was to try and compare these haplotypes to a reference genome. We would normally use the complete reference genome CHM13, but CHM13 has a big inversion in this locus. So we used another sample instead from the general population. This is AG00732. And as I was saying, once we have a good assembly, we can use pretty much any genome as a reference. And here I'm comparing at the top, we have this population haplotype compared to maternal H1, so haplotype 1, and maternal haplotype 2 here at the bottom. And again, here it's the population haplotype. So you can see also in red and green, the defensive gene, where they are positioned and their configuration. We have very different ones. And again, here in different color, you can see the repeats, the segmental duplications. So you can see that H1 only has a few difference compared to the population haplotype that we looked at, just some deletion here in red and one insertion here in blue. But on the second haplotype, we see a very big inversion. And if you look at where the defensive gene are and the repeats, it's very clear that this rearrangement was mediated by these highly identical sequences. So we know that H1 and H2 will recombine during meiosis 1, and the resulting haplotype will be transmitted to the child. And this is exactly what we see. This is the Carina haplotypes, and H1 is a combination of the maternal H1 and H2. So there is still a part of the inversion that remained, and on this side you cannot really see it, but there is a region, there is a sequence that is missed in a deletion, like the configuration that we saw before. The paternal haplotype instead was just transmitted with very few rearrangement. I have a slide at the end if you want to see how it looked initially. This is very important because once we have this complete haplotype, we can also do something that is usually very difficult to see. So here I'm showing you the proband H1 compared to the maternal haplotype 1 and maternal haplotype 2 here at the bottom. And this is an identity plot. So basically, the more red you see, the more identical is the sequence. And through this plot, we can identify where the arrow point, the breakpoints of the recombination. So we can really see where the recombination happened. And here, you can clearly see that, again, mediated by a repeat, there was an event that led to this rearrangement. The breakpoint in the middle is something that is very interesting. We did not expect it because usually inversion suppress recombination, but we see this transition to a less identical sequence. And so we think this might be real, but now Glenis has generated many more data for the same sample. So we are in the process of generating a new assembly and try to validate this. That would be very, very interesting if it turned out to be real.
It was interesting because you've never seen that before.
Yes, usually you don't see a breakpoint in the middle of an inversion. You can see that basically from this haplotype, this sequence was transmitted and then is switched to the second haplotype. Usually the breakpoint is at the edge, as you see here and here. In the middle, we don't really see it. So we are cautious in saying that it's real. We want to do more validation, but it's an interesting observation, and maybe we can. It wouldn't shed some new mechanism that we were not aware of a very specific case of a known mechanism. Okay. Next part is on variation genome-wide. So another thing that we wanted to do is try to find the structural variation and single nucleotide variation that contribute to the phenotype of 8p heroes, and that they are not present at the 8p locus, but elsewhere in the genome. So this work was done by Yang, another postdoc in our lab. She works a lot on single nucleotide variation and SVs, and as well as another postdoc in our lab called Jadong. Jadong basically worked on this database from 285 control individuals. These are still from the HPRC and HSVC, and used long-read sequencing to call SVs. And he did a very good job at highly curating this database, removing all sorts of false positive that were due to genome assembly or to read alignment. And he's keep expanding this database, which so far is pretty much the most complete SV database that we have that we can use to filter benign variants when we looked at pathogenic cases. So this is the work, the workflow that Yang uses. Is very complex, but I'm happy to talk about it after the presentation or even after the meeting if you're interested in. But looking at Bina's family, you can see how powerful it is to have this method. So Yang called 25,000 structural variation from this genome. And then she tried to validate all of them with read alignment methods, and the number dropped to 19,000. When she used this database to filter all benign variation, so all structural variation that were seen in the general population and in an unaffected individual, the number dropped drastically to just a few hundred. And here is to give you a visual comparison. We really restrict the search of clinically relevant variants to just a few hundred. And this was a great problem before for short read sequencing and even long read sequencing, because you end up with so many variants that you don't know where to look at. And among these, Yang looked at the additional de novo SVs, and she didn't find any. This is kind of expected because we expect 1 or 2 structural variation per generation. So we already discussed the one at the 8p locus, and it's not unusual that we won't find any other one. But we did find some other variations. I want to stress that these are not clinical tests. These are just variants that we prioritize for further analysis. A lot of these genes, we don't know what they do or what their pathway that they influence. But we did find some few rare homozygous SVs, and 2 of them involve genes that were previously reported by the Safari Foundation. They are classified as Safari 2. So again, we really don't know what they do, but it was an interesting observation. Other class of variation that Yang looked at were those that were present at the 8p locus on the paternal allele. So you can imagine if the maternal allele has a deletion, any variance that is inherited from the father, it will be an hemizygous state, pretty much mimicking an homozygous variation. And so she selected these 1 SMV and 1 insertion in these 2 genes that, again, were previously reported as Safari 2 genes, and one of them was reported in a previous study. And of course, they are rare in the population. As well as these, also she looked at all sorts of single nucleotide variation across the genome, and we have a huge list. I think probably the most interesting here is this compound heterozygous variant, then again involved a Safari 2 gene that we can look at before. These, as well as before, is all the data that I just discussed. They will be confirmed with the new genome assembly, and we'll take it from there for further validation if these are confirmed to be true. As well as this one, I am also looking at other loci in the human genome. This is an example of a complex variation in the 16p11.2. This is a child that was affected and have a complex rearrangement. You can see here an inversion mediated by this segmental duplication, as well as a deletion here. And we are interested pretty much in any susceptible loci in the human genome. So we are expanding our project, including these, and hopefully we're gonna have more data from other 8p heroes and families. And with this, I'm gonna go to the conclusion. So in this case, for the first family, but we want to do this for multiple family, we want to characterize the 8p locus, describing all the structural variations, their breakpoints, and also trying to describe and understand the mutational mechanism that underlie the formation of this structural variation, and also investigate if there is any genomic architecture that predispose to this rearrangement. And also another aim is to identify variants that were missed before with short reads or microarrays, and they could modulate phenotype or that of other 8p heroes. And I didn't talk too much about it, or I didn't talk at all, but we have long read sequencing and they have methylation data. So we are also going to look at methylation differences at the breakpoints and all the genes that are involved in the 8p locus. And with that, I want to thank all the people that collaborated on this work, and of course, Project 8p, Bina, Karina, and her family, and all 8p heroes. And thank you for listening. I'm open if you have any question.
Thank you, Kumar. Go ahead.
I will ask a quick question. So from this first sequencing, can you generalize or can you infer anything on other 8p heroes genomes, or you need to really sequence every single one?
I think to describe to this level of detail the rearrangement, and especially where the recombination happens and how we transition from 2 haplotypes to the one that is inherited from the child, we need the sequencing. But also we can say that we constantly see for 8p and other loci that repeats mediate this rearrangement. So the configuration of the defensive gene is very important, I think. And also I noticed that in the population haplotypes, I didn't show it here, sometimes some of the defensive gene are missing, sometimes the defensive A or the defensive B are missing. So I think once we have more data and we compare it to the general population, we can generalize on what is the most at-risk haplotype, let's say. But for sure, we know that the segmental duplications and the defensive gene mediate this rearrangement because of high similarity.
Thank you.
I had a question about these rare variants. So like that protocadherin variant, you know, that was a compound het. Like how much does that need to be prioritized, right? Because we have, you know, protocadherins are at the synapse. You know, we can start to paint a picture where, you know, relin is also there. That's a distinct kind of observation coming from other data. Like how much are we supposed to think about these rare variants as potentially disease drivers? And yeah, it seems like it's going to be unlikely that another AP hero is going to have exactly that compound heterozygous state at that locus. So is that really just to be interpreted as an N of 1? Or yeah, I'm just wondering what to do with these observations of these rare variants that could make an argument that they're drivers.
Yeah, I think the driver will always be the 8p rearrangement. But once we have very good data on the phenotype, so deep phenotyping, and we can really see the shades of the phenotype, so traits that are slightly different between 8p hero to 8p hero, we can really start to pinpoint what variation could contribute to this difference. And I think that's how you should see that that compound that. The compound that basically will mimic an homozygous variation, probably disrupting the function of the gene. So if we see a difference in the phenotype between two individuals that more or less have the same phenotype, but they have some slight difference, maybe some phenotype are more severe, maybe some phenotype are less severe, we can start seeing this variation and how they contribute. As well, of course, as I said at the beginning, the driver will always be the 8p. And to characterize exactly how the rearrangement is and what gene does it involve, I think is very important, even more important than the secondary variation. But I would consider as modulators.
So this is highly speculative, right? So I don't mean it in a bad way, right? So, but in human genetics since like whatever, 15 years, just to be clear, right? So because not many people are trained in on this call in genetic test interpretation or clinical genetic test interpretation. What you just mentioned is just very speculative, right? So every one of our genomes have like whatever 10,000 rare variants, and they are often also in highly constrained genes, and you know that's speculative. What these lists which have been provided. If I would analyze your genome, I would find similar variants if I filter hard, even for functional and important protein regions. Just want to be clear because most people don't know this. As a geneticist, you know this, but other people in this call maybe not. So
yeah, I want to add to that actually saying that the 8p locus will always be the one we should look at first, and characterizing where the breakpoints of the structural variation happen, but also where the recombination breakpoint happen is going to be very important, because in that process we can lose some genes, and especially some of the defensive genes that are very important for the immune response and to the health of an individual in general. I think that is where we should look at. And as Dennis said, we saw a lot of variation that involves Safari2 gene, and they seem to be too many even. So I think not all of them might contribute to the phenotype, or even none of them. This is something that needs to be.
I mean,
with genetic diversity, right? So and just to add, and I think just to comment, so the hypothesis that other variants are contributing becomes even more valid if daughter, for example, would look significantly different than other people with the inversion duplication, right? So then, and one can rule out that there is some kind of medical history and therapeutics and whatever drugs which could explain this, right? So, but what we have often learned from many people with highly penetrant genetic disorders is that there's a lot of factors which can play into this, and it's really hard to really up-level modifying variants. It's really a big challenge in human genetics in general. And it's really hard to qualify which of the thousands of other variants, depending on what kind of how you set the filters, right? Especially if you're not a Caucasian, right? How you set these filters from different databases. And the structural ones are really, the 2,000 people is nothing compared to the diversity there is in terms of structural variations to qualify a variant which really contributes or not.
Yeah, I think my only point is maybe a contrarian interpretation, which is that you keep saying the AP is the driver, but that even that statement you have to unpack because there's many loci affected in the mega bases of rearranged chromosomes. So you can't say that all of them are doing something. We already see a spectrum in the breakpoints of the patients, and yet they converge on similar phenotypes. That already tells you that there is no one uniform minimal critical region. So my point is that what if you view the AP locus as the modifier and that these rare, potentially pathogenic drivers on other loci are potentially the primary driver? I just want to keep that interpretation open. And it's probably some kind of middle two-way street, but I would not say that.
No, so you can narrow it down, right? So the inversion duplication is one, subdisease among the AP disorders. And the homogeneity of the inversion duplication has been shown, right? So there is already more homogeneous clinical spectrum. To which degree the inversion duplication disease can be attributed only to that variant, I don't know, to be honest. But Tobias will show some stuff later on that gives us good hypothesis that this is a more homogeneous group. And of course, nothing is 100%, right? But since you don't see this type of genomic rearrangement in people without neurodevelopmental disorder, you don't. So you can attribute it as a more penetrant variant for sure.
Yeah, and also these are complex rearrangements, so we call it inverted duplication, but they will also be accompanied by a deletion on the side. In most of these cases, there is even a duplication just next to the main variant. So all of those can modulate the phenotype and make this slight change in the phenotype in different individuals. But all the variants that I showed were filtered by this very large database that we have now. So it's very good that we can now also look at rare variation that happened in a repetitive region, and also this control data set includes all sorts of ethnicities. So we are able also to filter that and see the frequency in any population pretty much.
Okay, thank you all. I wanted to give Tobias some time. No, I know we have like a little under 20 minutes left. So Tobias, if you don't mind sharing screen.
All right. So yeah, thank you very much for having me here. Although I have probably a little bit less time, that's not a big issue because I think a lot of like the introductory part, which I've planned for this presentation, was pretty much discussed in detail here. So I can probably jump or only discuss some of these parts a little bit more fast than originally expected. So what I will be talking about is how the genotypes or how the specific CNVs can be linked to the clinical phenotypes in 8P disorders. And this is just meant as a progress update. So this is like ongoing work, which we will finish in the following months. So just to give a little bit of background, I mean, we discussed this comprehensively in the past minutes, but I just really want to highlight the high level of clinical variability in chromosome 8p related disorders. So here on the right, you see, for example, an image of a publication from a cure 4 years ago, where you basically every column presents a different AP hero, so a different individual with an AP disorder. And the color codes just indicates whether a phenotype is present or not. So every row presents a different phenotype. And just by like the structure and the color coding, you can really tell that AP heroes can look very different, and there's a high heterogeneous presentation of symptoms among all of the individuals. And this in the end makes it difficult also to predict the clinical course once you have the genetic diagnosis, and also eventually to optimize the treatment. adding to the discussion which we had before, I just want to highlight that on top of all the genetic factors which can impact the clinical variability of an individual, which means the CNV in 8P itself, other variants, there are a lot of like additional factors which can actually play a critical role for the clinical phenotype of individuals with 8P disorders, which can be access to therapies. So it can make a huge difference whether you start with speech therapy at the age of 3 months, or if you have never access to speech therapy at all. Like general environmental factors, then of course medications. A lot of medications and drugs have off-side effects, and the timing of the medication when you started the medication can in the end have a really large impact on the disease course, or like prolonged hospital stays for whatever reasons. They all can play a role, and with that, even an individual which might be identical, like in a theoretical sense, can present still different just based on these other factors, which can be environment, medications, and access to therapies. Just to keep this a little bit in the back of the head when we do like genotype phenotype analysis, that we always expect some variability which cannot be explained just by the genetics alone. Now, for CNVs, we have like a particular challenge in particular when we compare this to like monogenic disorders, as for example, copy number variants, which we observe in individuals with AP disorders, can span not a few genes, but up to hundreds of genes. And with that, we have like many different potential driver genes, even if we ignore the non-coding elements and everything like that. We still have like hundreds of genes which could potentially drive or at least modify the phenotype in individuals with AP disorders. And in the end, the breakpoints can vary, as we have learned also in the last talk. And each patient is like unique in its like genetic, in the presence of the genetic variants. Then we have like dosage dependent effects, which are linked to the copy number variants. And on top of all the genes, which might be drivers and which will be also the focus of this talk, there are also like a lot of non-coding regulatory elements, which can also play a role and have an impact on the clinical phenotype. Now, to give a little bit of background, this is not like the first effort to bring some or shed some light into the landscape of 8P genotype phenotype associations. So there have been a couple of publications in the past which describe, even for the different subclasses, that there is always a shared core of neurodevelopmental disorder features, which include some delay of speech, hypertonia, and this is seen across like many of the individuals with AP disorders, even a little bit ignoring the different types of CNVs which we can observe in AP heroes. On top of that, we see some patterns when it comes to the clinical burden in general, where there's a trend established that in particular individuals with inverted duplication and deletions seem to present clinically a little bit more severe than those individuals with only a deletion or only a duplication, always keeping in mind that these CNVs can vary in size by a lot, which can also impact the severity of the clinical presentation, as the size of the CNV is typically associated here in this case with the clinical severity, so very generally spoken. And then if we move or we zoom in this whole picture, we also see gene-specific traits. So for example, it has been previously suggested that a modification in GATA4 is highly associated with congenital heart disease. However, all of these previous studies, they have like a limitation that you need a certain amount of individuals to systematically assess the association of like a CNV class or a specific gene to an 8P related phenotype. And since this was missing, the goal of this project was to explore this now having access to the Project AP registry. And I think the registry itself was presented here a couple of times. It's just as a small recap. It's a registry organized by the Project AP organization, and it's a global natural history study, which is based on several surveys which are disseminated, and the answers of these surveys are incorporated and present the foundation for the work which has been done here. And I just want to highlight there is another survey which was started a couple of months ago, the organ system survey, which I think has the most phenotypic detail currently assessed. And with this survey being distributed more and more and information being collected from more and more individuals, this will also enable to further refine this analysis and provide access to a more granular analysis in the future, both on CNV level, but also on individual gene level. For this current state of this project, we identified 107 individuals where we had phenotype data available and the specific genetic breakpoints being characterized. And in the end, we are looking at 97 different phenotype traits, always in a binary sense. So just asking the question whether an individual has hypertonia or has not, but currently not yet looking at the severity of the presentation, whether it's a mild version of hypertonia or a very severe one. However, as mentioned before, with the periodic updates, we will increase the cohort size, increase the power in our analysis continuously, and also include more phenotypes. So this is an overview of the genomic architecture of the AP individuals with AP and rearrangements, where we are basically looking at, in the first step of this analysis, in 3 major classes, which are either the AP individuals with inverted duplication and deletion, which we see here on the left side, the AP deletions in the middle, and the AP duplications on the right side. And we really see that the cohort sizes really differ among these 3 groups. And what I really want to outline is that the size of the CNV differs dramatically. So every bar here represents the size across the along the chromosome 8 position, and you see that while there are a couple of CNVs, and I will discuss this a little bit further in a second, while the size of a couple of CNVs is like very narrow, others affect like tens of megabase pairs with a single CNV. Now, when we look in these individual groups, what we did is we calculated an enrichment of phenotypes to identify phenotypes which are more prevalent or more frequent in a specific subgroup of CNV disorders. So for example, here, for the cohort of AP individuals with inverted duplication and deletions, we looked at 97 different phenotypes and then try to identify phenotypes which are more present in this particular group of AP heroes compared to all the other individuals with AP, with also AP disorders, but which are only deletion only, duplication only, or some other version of a chromosome AP disorder. And what we identified here for the group with inverted duplication and deletions is that in particular, hypertonia, some behavioral issues like an individual who did not even make meaningful gestures or imitate the behavior of new individuals is enriched in this group of individuals with inverted duplication and deletions. While we see an opposite pattern, for example, when we look at individuals with AP deletions, where in particular microcephaly was, which was also previously described, was confirmed to be specific for this group of AP disorders, together with a couple other problems which stand out, like the problems of balancing and yeah, in general, like the motor impact of individuals with AP deletions. Finally, when we look at the duplication-only cohort, so individuals who only have an AP duplication, but again of various sizes, what we observed is that they had less an impact of motor skills, why the motor skills were more likely to be normal or in the range of normal compared to other individuals with 8P genomic disorders. However, they had still some issues, behavioral issues, while not reacting to the name, having poor eye contact, or some lacks of social communications. Now, the issue is when we just compare these larger CMV classes, is that we miss the opportunity, or we group individuals together which are genetically very different. And just to make the point here, if we look at the individuals with the deletion only, and we take the left group here in this orange box compared to the right group here, we see that all these individuals have only a deletion, but there's basically no overlap between these deletions. So they are basically deletions in the chromosome, or in the 8P part of chromosome 8. However, there's basically no overlap, meaning that the genetic variant is like the genomic region which is affected in these individuals is very different. And that said, the idea is to not only look at like these broad CNV classes, but to look in more detail on the individual genes which are affected by each patient, which can be very unique to each patient-specific genotype. Another thing which I want to highlight here, and this is also currently ongoing, is that in particular for these very small CNVs, in order to clean up the signal and also to confirm the classification, what we are applying is some kind of advanced versions of the CNV classification guidelines, which were originally proposed by Riggs et al. 5 years ago, to confirm that even like the small CNVs are pathogenic. While for the larger ones, it's often kind of implied, in particular, if you have ones which span like 20, 30 million base pairs. We have a couple individuals which are with 8P related disorders who only have like CNVs of the size of less than 1 mega base pair. And here we applied some advanced classification methods to ensure that each of these individual CNVs is indeed pathogenic and can be considered as the cause or as the driver of the clinical phenotype. Now, finally, how do we do the annotations on the gene level? What we did is we annotated for the first 70 million base pairs the genes and also a couple other non-coding elements. This is basically what we see here in the orange box. For all these genes and elements, we annotated then from various sources all kinds of annotations about gene disease validity, whether the gene or the non-coding region has been established with any disease already, whether they are known to be dosage sensitive, whether they overlap with a lot of individuals in the cohort of the Project 8P registry. And by incorporating all these factors, and I don't have time to go into detail like what exact filters we choose, we identified 39 genes which we call a potential disease modifier, which now in the next step, what is currently ongoing, where we will identify or test these genes, whether we find significant association with individual genes to certain phenotypes which are frequently observed in AP heroes. Now, finally, I just want to highlight how these data can be disseminated, as this plays always a crucial role in our lab. The second part of the project, which I think we presented here also a couple of months ago, is to integrate these results into the AP passport, basically enabling patients to get insights based on the individual genetics, very tailored to the patient, where the patient passport in general is meant as a personalized, really patient-centric tool, basically meant to summarize both the genetics and the clinical data, and with that make it accessible in an easy way, also easy language, to both like caregivers, but also clinicians who are not well trained with AP disorders. And by incorporating the work which I presented in the genotype phenotype insights, one can make it even more tailored to a specific patient by looking, for example, at the individual genes which are affected by the specific 8p CNV. And with that, I would just briefly summarize. So we can see that the CNV type matters. We can identify phenotypes which are associated in specific CNV types. However, this is not sufficient to explain all the possible phenotypic heterogeneity. And this is why gene level analysis are required and are currently ongoing. And I would just want to highlight here that this is anyways ongoing work, which will be, as mentioned before, incorporated into the AP passport, and which will gain more and more statistical power while novel phenotypes are included, but in particular as the patient cohort grows and the answers of the surveys are coming in over time. And with that, I would like to acknowledge everyone who contributed to this project so far, in particular the team around Project AP and Dennis providing support for this project. I'm open to take any questions.
Hello, hi, thank you for your presentation. I just wondered whether you can add another layer of information beyond the clinical data. Can you add maybe cellular characteristics from fibroblasts or other cellular models from the AP heroes, and whether that can build it into the whole overall picture that you are getting for this subgroup?
I think, I mean, I personally cannot do this, but I think this is in particular the goal of the Project AP Foundation, and this is why they take all these biosamples. That basically what we do, looking purely at the genetics and linking the genetics to the phenotype, is basically only one piece of information in this overall picture, which I think was also outlined beautifully in this whole roadmap. So I totally agree. This can only be part of the whole picture, and should be accommodated. And I think Bina can comment on this to all the efforts which the organization does in that direction to complete the
Dennis
picture.
aren't handling it all?
Well,
just a piece of, yeah, it's Nicoletta, I agree. It's a big team effort. I think it's that often, while we can even have like a roadmap of looking at each patient, separately and uniquely, and then also like trying to cluster together. It still seems like there's no like one lab or one organization that actually has even had maybe access to these types of data sets and how to actually make them synchronous and make sense together. So there's so many components to this. It's definitely the implementation of the roadmap that we put together is harder than it seems. And so Dennis will be like, well, it's common sense, it's straightforward, like we just need to do it. And I hear Dennis every time he says that, but then it's a monstrosity of an effort to make sense of it. So we're certainly trying, and I think that the fibroblast data that we're generating too has been important. So it's good that we're growing that biorepository and then also putting in the hands of investigators to sequence and do all the right cellular phenotyping. And then we're also I don't think any of our the modeling, the in vitro modeling, the cellular modeling folks are on this meeting today, but there are some efforts to then differentiate further into NPCs and different neuronal subtypes and then get data from that and compare.
Yeah.
I know we're at the end of the call, but again, I wanted to thank Kumar. Thank you, Tobias and Dennis, so much for just, putting your heads together on this. I know in sharing the interim progress, I think is really important too, so that everyone knows what each of us are up to, and hopefully then you can just feel comfortable reaching out to each other directly, which I think in the spirit of collaborating, especially in these difficult times, is going to be really important. So yeah, feel free. You all know each other's names. you don't necessarily need Project AP's team now, I think, to just connect if there's questions. So next roundtable is Monday. I know we try to do these once a month, but we're back to back just because scheduling and making sure. So really hope that you all can join us for that. And then we'll make a decision about the context of this conversation, whether we share this with our families or just keep it amongst our research community and if you have any thoughts on that, feel free to just send us a quick note. Okay. Thank you all. Have a great rest of your day and week.
Thank you.
Thank you.
Bye-bye.
Bye.
Thank
Be
you.
well. Take care.